Hi, hello everyone. Again, a warm greetings to everybody joined with us today for the sixth session of this international virtual conference on frontiers in biological research, which is organized by the Department of Botany in St. Joseph's College, Trichy, Tamilnadu, in association with the Biomix team. Today, for the sixth session of this international conference, we have another eminent scientist, Dr. Gaurav Zinta, who is currently working as a senior scientist in the biotechnology division at CSIR-Institute of Himalayan Bioresources Technology, is with us today, and he will be delivering his talk in this session on the topic on integrative view on plant responses to changing climate. On behalf of the Biomix and St. Joseph's College, Tiruchirappalli, I extend my warm welcome to you, Dr. Gaurav Zinta, and also I thank you for accepting my invitation for this talk. Today, our speaker, Dr. Gaurav Zinta, will deliver a comprehensive view on the efforts of global climate changes on various plant species and also a multi-scale approaches spanning from plant physiology, biochemistry, transcriptomics, epigenomics, and mathematical modeling were implemented to decipher plants' responses to climate change. And also, he will be interpreting the results obtained on Arabidopsis, European grasslands, and cereal crops with the various parameters such as the first is high CO2 mitigates the impact of combined high temperature on drought risk, on growth and photosynthesis. Second is short and long-term exposure to climate extremes impact primary metabolism differentially. The third is repeated heat stress exposure induce transgenerational adaptation, where DNA methylation plays a crucial role. Fourth is nutrient quality losses are less prominent in grasses than legumes under climate change conditions. Fifth is grasses and legumes execute unique protein accumulation biochemical strategies to cope climate stress. And sixth one is crops belonging to different photosynthetic function groups, that is C3 and C4, respond differently to stressors under high CO2. And also, he will elaborate the overall mechanistic insights on plant responses to global climate change and the importance of integrative approaches to explore species-to-species responses to global climate. At last, Dr. Gaurav Zinta will also provide a glimpse on the role of modern plant breeding tools such as CRISPR-Cas9 to engineer plant rights. Now, I hope you all are eagerly waiting to listen to his lecture. Before listening to his lecture, we the listeners must know the academic biography of a personality, one who is delivering a lecture. For this, I request one of our organizing secretaries, Dr. Anusha Satyanarayanan, Assistant Professor in the Department of Botany from St. Joseph's, to introduce our today's speaker, Dr. Gaurav Zinta, to the audience.
Thank you, Mr. Parthasarathi. I'm extremely happy to introduce Dr. Gaurav Zinta. He's currently a senior scientist in the biotechnology division at CSIR-Institute of Himalayan Bioresource Technology, Palampur, Himachal Pradesh. He availed 2 fellowships to pursue his PhD from the University of Antwerp, Belgium. After completion of his PhD, he worked as a postdoctoral fellow in the same university. Also, he received a postdoc fellowship from the Chinese Academy of Sciences to work at the Shanghai Center for Plant Stress Biology, Center of Excellence in Molecular Plant Sciences, CAS, China. The key areas of his research interest include integrative molecular plant stress physiology, climate change biology, genome editing, epigenetics, and chemical genetics. During his PhD, he worked on how plants respond and adapt to global climate change. Arabidopsis and European grassland species were used to elucidate the impact of climate changes. Specifically, he used multi-scale analysis to decipher plastic and adaptive plant responses to combinatorial and long-term stresses. Such integrative analysis is crucial to get deeper insights into plant processes and develop climate change adaptation strategies in the future. During his postdoc, he worked on the aspects of plant stress epigenetics and chemical genetics. Also, he used gene editing CRISPR-Cas9 to manipulate gene function to develop plants best suited to grow under harsh environmental conditions. So far, he has published 31 peer-reviewed articles, 2 book chapters in top-notch journals, including Nature Communications, Global Change Biology, Journal of Hazardous Materials, Biotechnology Advances, Trends in Plant Sciences, etc. He participated in several training courses and conferences across the globe, including Germany, Ireland, Poland, France, Belgium, Netherlands, etc. He is a member of editorial boards of several journals. He is a featured editor of Molecular Plant and associate editor for Plant Environment Interactions and CABI Agriculture and Bioscience. He is serving as a review editor for Frontier in Plant Science and Frontiers in Genetics. He also participates in several science outreach programs for select With this, I conclude the introduction of Dr. Gaurav Zinta, as it is a very big one. I have just shortened all those things, and now the platform is yours, sir.
Hello. Thank you, Partha and Anusha ma'am for the nice introduction, detailed one. Now I would like to present my work. Let me share if you can see it. Yes. Yeah. Are my slides visible?
Yes, sir. You can proceed ahead.
Yeah. Thank you all for such a nice, wonderful introduction. So today I'm going to present my work with the title An Integrative View on Plant Responses to Changing Climate. Most of the work I have done during my PhD at University of Antwerp, Belgium, and at Shanghai Center for Plant Stress Biology at Chinese Academy of Science in China. So I recently, last year in May, joined as a senior scientist at CSIR-Institute of Himalayan Bioresource and Technology, Palampur, Himachal. This is a brief introduction, and I did my PhD with 2 research groups. One was plants and ecosystems. Another one was integrative molecular plant stress physiology. My promoters were Professor Ivan Janssens and Professor Hanaza. Then I got a fellowship from Chinese Academy of Sciences to pursue my postdoc at Shanghai Center for Plant Stress Biology with Professor Xiangkang Zhu. He's also a distinguished professor at Purdue University, USA. So I worked there for almost 5 years, and this was my brief academic journey. So now I'm coming to the topic. Plants are sessile in nature. They cannot move. They are, confronted with different kinds of abiotic stresses. They are, these abiotic stresses challenge plants to the level of their survival. So there are different kinds of, soil-borne stresses like flooding and drought. There are aerial stresses like high temperature, cold, ozone, UV, highlight, heavy metal, and salinity again are, soil-borne stresses. So the idea of this slide is to show that in nature, in their natural habitat, plants are challenged with different kinds of abiotic stresses. So how the plants respond and adapt to high temperature stress? This is a key aspect or a priority. This is a key aspect of global climate change. So if you see here, high temperature stress leads to yield reduction, reduction of quality traits, reduction of plant growth, inhibition of seed germination, water loss, inhibition of photosynthesis, and oxidative stress. So what kind of defenses plants trigger in response to high temperature? There are different physiological, biochemical, and molecular responses, which include change in the leaf orientation or leaf rolling, transpirational cooling, alteration in membrane lipids, the production of asthma-protectants and antioxidants, and expression of different kinds of stress-responsive proteins. So oxidative stress, is a central hub, or it is a central hub triggered by different kinds of abiotic stresses, which include halide, wooding, herbicides, senescence, ozone, drought, pathogens, heat, and heavy metals and salinity. So, what are reactive oxygen species? These are highly reactive oxygens. These are very, reactive species, which include hydrogen peroxide, cyclet oxygens, superoxide anion, hydrogen peroxide. So in this chart, in this figure, I have categorized them on the basis of their reactivity, where hydrogen peroxide is least reactive in nature as compared to hydroxyl radical. So to cope with these reactive oxygen species, various antioxidants, molecules, and enzymes come into place, which are ascorbate and glutathione, phenols, and tocopherols are antioxidant molecules. There are different kinds of antioxidant enzymes, which include superoxide dismutase, catalase, ascorbate peroxidases, etc. Under optimal growth condition, there is a balance between reactive oxygen species and antioxidants. So, however, when stress is severe, the level of reactive oxygen species increases, and antioxidants are unable to, scavenge these species. So, which leads into oxidative stress and oxidation of lipids, proteins, and DNA. So, as I stated in the beginning, this talk will be primarily focusing on climate change. So, what is climate change? Climate change has mainly three components: rise in the atmospheric CO2, which leads to earth warming and increase in the frequency and intensity of drought episodes. So, International Panel for Climate Change has given three emission scenarios of CO2. One is low emission scenario, which is shown with a blue line, and there is a middle emission scenario for CO2, which is represented in green line, and there is a high emission scenario of CO2, which is shown in red line. So in these all three scenarios, what we can see is there is increase in the Earth's temperature by the end of this 21st century. So this is how the global map looks in terms of temperature. We can see that the global Earth temperature will be higher in the future. So in this presentation, I'm going to split my content into four parts. First is, how does future climate CO2 alters stress responses? So here I will talk about the combination of heat and drought together with high CO2. Number two is how plants evolve under extreme heat. So here I will be talking about the transgenerational effects of heat stress. Number three, role of epigenetic components in climate change adaptation. So as we know, genetic changes are long-term and random in nature, but epigenetic changes which involve alteration in the DNA methylation patterns or histone modification can act as a fuel for adaptation. Number four, what kinds of smart technology are available to us, and I will be basically describing CRISPR-Cas9 genome editing and epigenome editing tools. So this is the first experiment which I really started during my PhD with Arabidopsis. As we know, Arabidopsis is a weed. The life cycle of this Arabidopsis is 2 to 3 months, and we have a lot of genetic resources available on this plant. A lot of mutants are available. Transformation is very easily, so it is very beautiful system to work with. So in this experiment, we treated Arabidopsis with we grew Arabidopsis under controlled environmental conditions in 4 conditions. One was a control, which was receiving both optimal water supply and temperature and optimal levels of CO2, which is 360 ppm. Another one was high CO2, which is 730 ppm of CO2 to simulate the future climate. Third one was combination of heat and drought, which I'm representing with the HD in triangle. And another one was the fourth one, heat and drought combined with CO2, which I have represented with the red triangle. So these are the four conditions: control, CO2, heat and drought combined, heat drought combined with CO2. So I set up an experiment and I did a lot of physiological and biochemical analysis. So basically, this graph represents the sampling time point at 32 days of growth, sowing, and 36 days of sowing, 40 days of sowing, and 45 days of sowing. So at different harvesting points, we did a lot of physiological and biochemical analysis. We also did microarray analysis to profile gene expression patterns. So how does our climatic treatments affected the growth of Arabidopsis? So if you can see here is CO2 plants were really bigger in size as compared to the control plants represented with C. And heat and drought significantly reduced the growth of CO2 plants or control plants. So when CO2 was combined with heat and drought, we observed a sort of mitigation of stress response. So this is basically a quantitative representation of the data where we are quantifying rosette biomass and in gram fresh weight. What we can see here is there is a decline of 76% under heat and drought. However, this decline was only 52% under heat and drought combined with CO2. So this slide clearly gives us an idea that CO2 mitigates the effect of heat and drought in Arabidopsis. So this was an exciting observation. We wanted to look why CO2 is really protecting plants from heat and drought. So we performed genome-wide transcriptional analysis by using microarray. So microarray is a technique by which the whole genome level changes in the transcriptome can be profiled. So what we see here is heat and drought with heat and drought, 3,640 genes were differentially expressed, and with heat drought combined with CO2, 2,841 genes were differentially expressed, and majority of the genes were commonly shared in these treatments. So we perform the MapMan pathway analysis. So basically, this is a bioinformatic approach to map the biochemical pathway at the level of gene expression changes. So you can see here in this figure, there are different kinds of pathways which were really altered by heat and drought. So the red color represents down regulation of the genes, and blue color represents the upregulation of the genes. So these blocks basically represents different different classes of the genes from one pathway. So if you can see from here that photosynthesis or light reactions were strongly downregulated, and this patch is really red, and other pathways like sugars, lipids, and amino acids were altered during heat and drought combination. So we also performed the similar analysis under heat, drought, and CO2. And when we, observed the differences, we could see that heat and drought, there were few genes which were upregulated in the photosynthetic pathway. So these slides give us a view that heat and drought in combination with the CO2 is mitigating the effect of, on photosynthesis at the transcriptional level. So we come up with some physiological analysis. So we measured photosynthesis, we measured chlorophyll fluorescence. So basically in photosynthesis, we can see that under heat and drought, there was strong decline in photosynthesis, but in heat and drought combined with CO2, the photosynthesis was less, decreased. The same is true with the Fe over Fm. With heat and drought, there is a strong decline in Fe over Fm ratio, but in heat and drought combined with CO2, the decline was less. So it gives us an idea that CO2 mitigates the effect on photosynthesis. So we also profile different kinds of oxidative stress markers. We quantified hydrogen peroxide levels. As I said, it is a reactive oxygen species. We also quantified protein carboxylation status, the protein carbonyls. So what we can see here is heat and drought resulted in a significant increase in the reactive oxygen species. However, CO2 in combination with heat and drought resulted in the decline. And the same is true with protein oxidation. So next, we quantified the level of antioxidant. I quantified, total antioxidants, and the total phenolics. So what we can see here is the total antioxidants were higher in case of heat drought combined with CO2. So what is the take-home message from this slide is higher accumulation of total antioxidant and phenolics at elevated CO2 would explain the stress mitigation effect of CO2. And we have quantified other, antioxidant, but for the sake of simplicity, I'm not showing it here. So then we, switched into the cause. Basically, hydrogen peroxide is produced. Photorespiration is one of the key pathway for the generation of H2O2, and photorespiration is a pathway which occurs in three organelles: chloroplast, peroxisomes, mitochondria. So ribosco is a bifunctional enzyme which can fix CO2 and can also react with the oxygen, and it depends on the concentration of CO2 and oxygen in the cell. When it reacts with CO2, it fixes it, and the Calvin cycle takes place. But whenever there is, high oxygen level, especially during the stress episode, glycolate is produced, and ultimately glycine and serine are produced. So the production of glycine and serine can be used as, the biochemical markers of photorespiration. So in our case, we quantify glycine and serine ratio, and what we can see is under heat drought plus CO2, the glycine serine ratio was lower, which gives us an idea that less reactive oxygen species, or we can say hydrogen peroxide, was produced because of low photorespiration taking place during high CO2 concentrations. So now I'm coming again to this previous slide where I showed you that different kinds of sugars, lipids, and amino acids were changing. So we profile these metabolites also. So what we can here see in this plot here is this is the principal component analysis of all the metabolic data. We can see that short-term exposure of 36 days as well as long-term exposure of 40 days are clearly in different panels. So giving us an idea that there is a differential impact of short and long-term exposure of heat and drought in the metabolic profile. So when we did heat map cluster analysis, what we can see here is that sugar is this plant. So we leveraged our experiment to European grasses or grassland species. So we studied the impact of climate change on Lolium perenne and poa, and legumes like medicago and lotus. And these are two papers which came out of this work, and which suggest that grasses activates glutamate pathway for protein accumulation, while legumes activates ornithine pathway for protein accumulation. And also, we observed that quality losses are less prominent in grasses as compared to legumes. So what I want to demonstrate from this slide is that species-specific responses exist. So we have to be careful when we are making generalizations from the studies which we are doing. So now I'm coming to the next experiment, where we were trying to understand the long-term effect of climate change, or you can say heat stress on plants. So I did the experiment with the Arabidopsis. We started with one single plant. In generation 0, this G0 represents generation 0. So we grow plants under optimal growth condition for 4 generations under 21 degrees centigrade. We also grew plants for under heat stress conditions, and this is the 38 degrees centigrade for 10 days for the heat exposure duration in each generation. So in every generation, we selected plants for seed biomass and did the selection for 4 generations. And at the end of 4 generations, we did the reciprocal transplantation experiment where we grew plants selected from heat, control selection, selected plants under heat, as well as heat selected plants under control conditions. So what we could see from this experiment here is, in terms of fitness, this is the axis of control selected plants, how they are behaving under control and heat stress. So we can see here is plants grown under heat stress result in significant decline in the seed biomass. However, in heat selected plants, this decline is less as compared to control selected plants. This clearly indicates that heat selected plants, or the plants which were repeatedly grown under heat stress, have a fitness advantage. So what are the reasons for this? Then we again did genome-wide transcriptional analysis, and we compared heat selected plants with control selected plants, and control selected plants exposed to heat, compared it with control selected plants, and heat selected plants which were exposed to heat were compared with control selected plants. So these are the different genes which were differentially expressive. The most interesting pattern which we could see here is the comparison between heat selected and control selected plants. So there were 31 genes which were constitutively overexpressed in these genes, in these heat selected plants. So when we did the functional analysis or geoanalysis of these genes, we could find they belong to different functional categories of oxidative stress, response to chitin, cold, wounding, light intensity, mechanical stress, or reactive oxygen species. So we also performed whole genome DNA methylation. We also performed whole genome bisulfite sequences to profile DNA methylation patterns, alteration in DNA methylation patterns. So I'm not presenting it here, but but what we can show from this slide is that repeated exposure to the heat stress results in the upregulation of stress responsive genes, and this was linked with the alteration in DNA methylation patterns. As I said, that epigenetics plays a very important role in stress adaptation. So what are epialleles? So epialleles are genes which are similar in their sequences, but they are different in their methylation status, and they are inherited from one generation to the another. So their genomic sequence is similar, but they are different in epigenetic status, especially DNA methylation. So in this work, we categorized the epiallele in Arabidopsis. We were working with 2 ecotypes, natural strains of Arabidopsis. One was called 0, and another one was C24. So when we compared the whole genome data of these 2 accessions or these 2 natural strains of Arabidopsis, we could find different methylation regions. So for instance, in this C24, this region at line 1 retransposition was really methylated. So when we did the crossing of these 2 ecotypes, this methylation region was inherited to the next generation. What we could, what we could infer from this figure is that these methylation regions could be inherited and to the next generation in the reciprocal crosses which we made. So we were really interested what is the function of this region in Arabidopsis. So we opted a fine mapping approach. We could map the gene, and this gene we named it as NMR19, and then we also studied different other 144 accessions of Arabidopsis to see the methylation status of this particular gene in Arabidopsis. And we could find that in 4 ecotypes or 4 natural strains, this was unmethylated, then it was methylated, then it was in 78 ecotypes, it was even deleted. So this is how the this region was methylated in different accessions in Arabidopsis. So then we started look, whether there are some phenotypic differences related to the methylation pattern of this particular apially. So we could identify that this methylation pattern in NMR 19.4 is negatively associated with the leaf senescence. And we also could figure out that this is linked with the hot climate of the ecotypes. So the idea from this presentation is this, this figure is that methylation status of particular gene can regulate any phenotypic trait, and this phenotypic trait can be linked with the climatic adaptation. So the overall conclusion so far is that elevated CO2 mitigates the impact of climate change, and short and long-term exposure to heat stress alters the metabolism distinctly. Repeated exposure to heat stress improves plant fitness, and epigenetics is involved. When we looked into the DNA methylation pattern, they're involved in climate change adaptation. So now I'm taking you to a different, little bit different area where what are the technologies we have to engineer plant or to engineer climate resilient crops. So we have a very versatile tool which we call as CRISPR-Cas9, which we can use for gene editing and epigenome editing in plants. So what is CRISPR-Cas9 genome editing tool? So as you are aware, this the developers of this technology got Nobel Prize last year, and CRISPR-Cas9 is a tool by which you can create mutagenesis, or you can insert a gene, or you can even replace a base. So it can be used for mutagenesis, for knock-in, or precise base editing. For instance, CVE editor is used for C to E replacement of a base, or A to G replacement of a base. Likewise, ABE editor can be used to replace A to G or T to C. So this is, as I said, a versatile tool. So we implemented this tool to engineer herbicide resistance in crops. So this is an example of one herbicide we were working. We call it as mesotrione. Mesotrione is a herbicide which inhibits HPPD gene. So HPPD is basically a 4-hydroxyphenyl pyruvate dioxygenase, which is a crucial enzyme, and if we inhibit this enzyme, the plastoquinone and the tocopherol is not synthesized, and it results in the bleaching of the plant. So basically, when we spray herbicide mesotrione, so this inhibition of this enzyme takes place, and the bleaching occurs. So it's a broad spectrum herbicide. So we wanted to, take an advantage of CRISPR-Cas9 to engineer crops which can make resistant forms of, HPPD gene. So this is an example of rice and soybean, where we could see that gene-edited plants in rice and in soybean were resistant to these herbicides. So then, as I said, CRISPR-Cas9 can be used for epigenome editing as well. So if Cas9, dCas9 is fused with methylase. So basically, methylases are enzymes which recruits or which methylate a gene, and demethylases are enzymes which help in the erasure of methylation pattern. So when you fuse dCas9 with methylase, we can have enhanced methylation pattern in the promoters. When we fuse dCas9 with demethylase, so basically in the promoter region of the genes, there will be erasure of the methylation pattern. So as we know that methylation patterns can regulate gene expression, so we can use CRISPR-Cas9 to modulate the gene expression pattern without changing the sequence of the gene. So we did epigenome editing in tomato, and if you can see here, this is control plant, and this is red represents different CG methylation patterns, blue represents CHG methylation patterns, and green represents CHH methylation patterns. So when we use the TET demethylase, as I said, demethylase will erase the methylation patterns. And what we can see in these transgenic lines, the methylation was removed, the methylation was erased, and this nicely fits with our, expression of the gene expression pattern also. So the idea from this slide is that epigenome editing can be achieved by fusing dCas9 with methylase or demethylase. So, as I said, I recently joined CSIR-Institute of Himalayan Bioresource Technology. We are mainly interested in how plants of the Himalayan region respond and adapt to climate change. As Himalayas are represents fragile ecosystem, so it's imperative to work in this direction. So we use an integrative approach to understand the molecular mechanisms. So these are my PhD students, Komal, Pravesh, Madhushri, and Vidhi. So I thanks all my previous collaborators, my mentors. Thanks to CSIR IHBT, and thanks to all of you for your attention. I'm open to the questions.
CRISPR, Cas9 to engineering, plant trials, all these things are quite interesting. And we have a few questions to make this session more interactive with you. Shall we start?
Yes, sure.
Yes, sir. As you discussed the last questions, last part of that is dCas9 with combining with. Does it possible for every species? And
yeah.
Hello.
Go ahead, please.
Am I audible?
Yeah. Go ahead, please. You are audible. Go ahead, please.
Yes. I hope am I audible, sir?
Yes, you are audible.
Hello. Yes. Shall we proceed with the same question once again?
Yeah. Go on.
Yes, sure, sir. So there are two different mechanisms in dCas9 methylase and dCas9 recruiters.
Yeah.
That is dCas9 methylates and demethylates. When we combine two different things, when we combine dCas9 with methylase, it stops the transcription without altering the gene sequence. When we combine dCas9 with demethylates, it enhances the transcription. These are two different concepts that you have delivered.
Yeah. Yes. So I will answer your question. So basically, what happens if you see if you compare the methylation patterns of a gene, if a gene promoter is methylated, it will result in gene silencing. If a methylation promoter is demethylated, it will result in the expression of a gene. So we can use dCas9 system fused with methylase or demethylase to erase or methylate a promoter without changing the sequence of a particular gene. So basically, by altering the status of the promoter, the alteration in the gene expression can be achieved. This is what I meant from here. So basically, without any alteration in the gene sequence itself, you can achieve the expression pattern, alteration in the expression pattern.
Yeah, sure, sir. So I'll pass on to the next question. You have talked about broad range of IHBT herbicide that is used, a broad span of herbicides is used against plastoquinone and tocopherol. It has been inhibited by mesotretone, which has been is an enzyme which has been curbed by with the help of use of CRISPR technology.
Yeah.
In this mechanism, why don't we use Cas9 methylase or demethylase to inhibit the enzymes called HPPD?
Okay, that's a very good question, actually. So we have to understand one thing. Here, a herbicide is binding with a protein. So binding with the protein pocket and making it dysfunctional. You know my point? So this has nothing to do with the the expression pattern. So we have to engineer a protein in such a way that it retains its normal functioning, but at the same time it don't bind to the herbicide. So to create such kind of mutation, CRISPR-Cas9 was used so that we can get a variant of HPPD, which can produce, which can function, which can act, which can perform its normal function, but at the same time it don't bind to the herbicide.
Yes, thank you so much, sir.
Yeah.
Then coming to the happy allele, we talked about the two different species, we compared about the two different species of Arabidopsis, where we characterized the happy alleles. Happy alleles are that they have same sequences, but obviously they are different in their methylation process.
Yes.
In that, we have compared COLO and with that of C24. Whereby in C24, we have evidenced a huge number of methylated process, methylation genome.
Yeah.
And what about these methylations in F1 and F2 generations, sir?
Yeah. So as I said, when we made a reciprocal cross of COL0 and C24, because this region we found in C24. And we wanted to see, because methylation patterns are not always inherited. So only when methylation patterns are inherited, we call them as epi-allele. So when we cross them, these ecotypes, COL0 versus C24, we found that these methylation patterns were inherited, suggesting that this is a true epi-allele.
Oh, what we have found as a true epi-allele.
Yes. Otherwise, alteration in the methylation patterns you can find throughout the genome. Whether these methylation patterns are really inherited to the next generation or not decides whether it is an epi-allele or not. If it is inherited to the next generation, it means it is an epi-allele. If it is not inherited, it is not an epi-allele.
Yes, sir. What is in case of that plant C24 in that particular species of Arabidopsis?
You want to know what? The methylation pattern was more.
Yes.
Yeah.
In that species of Arabidopsis.
Yeah, the methylation pattern, we found more methylation in this particular region.
We shall pass on to the next question. So you have worked in two different plants, which is basically a model plant, Arabidopsis, and another plant, grassland plants as well.
Yeah.
What was your experiences in working with a model plant and a different plants like grassland species?
Yes.
What is experiences? How do you feel? Which one is comfortable to work with genomics level and other protocols in the experiments?
Yes, this is also a very good question. Actually, when we compare Arabidopsis, we call it as a lab rat of plant scientists. So working with Arabidopsis is very easy, I must say. Whether you want to transform it, whether you want to have mutants of Arabidopsis, there are different genetic resources available for Arabidopsis. So handling Arabidopsis is very easy. But working with grasslands, we don't have first genomic resources. We don't have the life cycle of these plants is also not short as Arabidopsis. So working at the molecular level, other than Arabidopsis, is a bit difficult. But with the advent of sequencing technology, now we can see rice as a model system also, as a cereal model. Medicago trunculata is also there as a model legume. So Brachypodium is also model grass to understand stress responses. So genomics has really helped us. But as we know, I mean, if you want to, transform or do gene editing, still, transformation protocols are not available for every species. So if you want to really pinpoint at the level of, genetics or do validation, it's still very hard to work on other species as compared to reptiles.
Yes. Yes. Next, coming to different question that whenever we trace a plant, whenever we trace a plant, we study about a plant, a proline accumulation plays a vital role among the rest of the amino acids. Still, there are newly found amino acids or solenocysteine and these things. But why we concern more about proline accumulation? In case of even in your research paper, you have correlated proline with the case of climatic conditions. Why it is so? It's very specific to proline aspect.
Proline, proline is a beautiful, molecule. So as I said, I mean, it acts both as an antioxidant as well as osmoprotectant. So meaning that it has the ability to scavenge reactive oxygen species, as well as when there is, a fluctuation in the level of, osmoticum. So it accumulates. So it can help you both at the level of osmotic adjustment as well as scavenging of reactive oxygen species. So this is crucial. I mean, when you, let's say in our case, when we were growing Arabidopsis to high temperature stress, so we can expect that reactive oxygen species can accumulate there. At the same time, we were combining it with drought, so we can, foresee, we can think that osmoticum of the cells can alter. So that's why, I mean, we looked into proline because it's a molecule, as I said, I mean, with both antioxidant and osmoticum properties.
Yes. Again, coming to the same paper that we have published, this proline concentration has been evidenced in the grasses. Have we performed the same experiment in the Arabidopsis plant?
Yes. In our case, I mean, when we compared, see, there are two things. Proline is produced by two biosynthetic pathways. So basically, when we were comparing grasses with legumes, we could see that no doubt proline was accumulated in response to climatic extreme. But they were using two different pathways to accumulate proline. So this is, if, for example, we would have been using Arabidopsis, so we would have, identified, okay, proline is accumulated. But when we use two different species or two different systems, legumes versus grasses, so we were able to disentangle the difference at the biosynthetic pathway level.
So can you explain last thing once again?
So I'm saying that when we were, comparing grasses versus legumes, we could, disentangle, the biosynthetic pathway of proline accumulation, because grasses and legumes were using different pathways to accumulate proline. The end product was accumulation, but the pathway opted by grasses and legumes was different.
Yes, fine, sir.
Yeah,
I shall pass on to the next question. Is something. So again, coming to the epi-alleles, C24, where we evidenced more number of methylation process in the species of Arabidopsis, that is C24.
What could you please?
C24. In that methylation, sir, does this shows any transfer of next generations? That is in F1 and F2 generation. That was my question.
Yes, I mean, this is what I have shown in the data also, because this C24 region, in C24 ecotype, this methylation region was higher. There was this region we found in C24. When we crossed, and this region was absent in col 0. So when we crossed it, in the reciprocal, cross, when we cross male, use, col 0 as male and, C24 as female and vice versa. So we could see that this region was inherited in the F1 progeny.
Okay, fine, sir.
Yeah.
Thank you so much for your patience in listening all my questions and answering all my questions. Thank you so much.
It's my pleasure.
Now I open the forum to the organizers or the panelists over here. If they have any questions, they can directly forward the questions to our speaker.
So, thank you so much for your nice presentation, sir.
Yeah.
As well as it is a current area. And now I have a small doubt. So, the methylation of NMR 19,4 negatively associated with the leaf senescence and the hot climates.
Yeah.
So usually the hot climate, the leaf are undergoing the senescence.
Yeah.
Whether they have any role in Himalaya nearby, because there is a hot climate is not always the profile. No? So is there any difference between these parameters?
Okay. This is about, yeah. Okay.
The second is, if the some of the crops which are subjected to the stress physiology, okay, whether any factors, the same crop is cultivated in the southern part, whether is there any impact on the metabolites generations or any other nutritive value difference?
Yes.
That's the two questions. Please.
Yeah. So it's a very good question, actually. If you see, I mean, in terms of crop, we always talk about stay green phenotype. Stay green phenotype is a phenotype of a plant to stay green under high temperature stress. So what happens under high temperature stress? It leads to the degradation of chlorophyll content or chloroplast. So leading into the senescence. So basically, under high temperature stress, plants plants die because of chlorophyll degradation. So, in our case, what we could see is this epi allele was negatively regulating these, chlorophyll biosynthesis, and this negative regulation was also linked with the hot climate. So, and coming into your second question, that whether there will be differences in terms of metabolic changes or nutrient profile comparing to the northern and southern part of India. Definitely, I mean crops which are growing under optimal conditions have a different metabolism. Plants growing in stressful conditions can have a different metabolic profile. If we compare the nutrient, I'm not sure. I mean, growing plants, how in temperate region will behave when growing into the southern part. So it depends on the species also. For instance, if we compare C3 crops versus C4 crops, so when they are exposed to high carbon dioxide temperature or high carbon dioxide or high temperature, the nutrient losses observed are more as compared to C4 crops. Like C4 crops, I'm saying maize. People have demonstrated that with the climatic changes, the nutrient loss in these C4 species is less as compared to the C3 crops like rice. So that's why people are projecting C4 crops as one of the future crops. Like maize is more beneficial as compared to C3, let's say.
Yeah, thank you so much, sir, for the presentation and being here, once more time in our platform. Thank you so much, sir, for that.
Thank you very much, Partha. Thank you, everyone.
Yeah, thank you so much.
So thank you. One second. Thank you so much for your active presentation and the present scenario, and also the way by which you delivered is very appreciable, and most of our staff members as well as our students community can absorb the stress physiology, and We are very honored, and on behalf of our college management and behalf of our botany department and all the staff members of my colleagues, those who are joined in our that is in our online. So I wholeheartedly thank everyone, and I express my sincere thanks to you, sir. Thank you so much, sir.
Thank you very much.
Thank you, Speaker, and thank you, the organizing secretaries. Rajanji, thank you very much.
Thank you, sir. Thank you.
Yeah, thank you. Bye-bye.
Thank you,
Good
sir.
night. Bye-bye.
Thank you, Partha. Thank you for the arrangements.
Thank you, sir. Thank you.
Thank you, sir. Churchill, sir. Thank you, Johnny, sir. Thank you so much for joining, sir.
Congratulations.